Hello microbiology learners, this is Professor Salm and I'm going to give you a quick and dirty screencast on how to navigate through the Marian Online 2 course shell and also tip you off to what you're going to need to complete for next week's class. So at the top of the course shell you've got a news forum, which there's nothing active at this time in the news forum. You'll receive notification of any posts that I put in the news forum. I don't use it very often, just to let you know. There's the syllabus here in a PDF form, so if you click on it, it'll download and open for you. So you have that, although I also gave you a hard copy in class yesterday. Then you've got the Microbiology Online Companion, which is, this first one is for those who purchased the ebook. For anybody, whether you purchase the ebook or the hard copy of the text, you have this freeware online companion, which I'm going to click on. Now I want to show you how to navigate through this freeware because there's just oodles of great tools and resources for you to become familiar with the subject of micro and as well as understand what kind of cool things are built into the text. So we have these links up here: careers in micro, supplemental case studies, which I may pull from, classification in prokaryotes. Well, let's see what that is, actually. I'm kind of curious. Nice, okay. So it's got all the geeky scientific Latin-y names. I'm actually going to steer you away from this particular that particular resource because I have my own little thing that I've established for you for prokaryotic classification. Recommended readings, virtual labs. I'll let you know if any of these become, I mean, you can cruise through them and see, but I don't plan on assigning anything specifically through them. What I am interested in, though, is this Chapter 1, the drop down for the chapters. You'll want to select whichever chapter is assigned. So I'm assigning chapters 1 through 4 practice quizzes to be completed for next week. And again, these are meant to sort of steer you through the text and kind of highlight some of the more important points in your readings. So for the practice quizzes, when you open them up, you get about 30 questions. And when you do those questions, I'm just going to go ahead and spam out a few answers here and show you what happens. Now, these, when you submit, you get a report that says how many you got correct or incorrect. And of course, I don't like this score. So ideally, I want to be able to reattempt. So you've got your correct versus incorrect, and you have your unanswered questions. So if you don't answer, you don't get any feedback at all. But then I'm going to go ahead and email these you can actually email them in an HTML format or text. And then I think if I pick text, no, I'm going to go ahead and stick with HTML. I've actually created an inbox just for this course. So if you email them to yourself, I'm going to go ahead and email them to my Marian email address. And also, I would recommend emailing to yourself and then also email to salmmicrobiology@gmail.com. So that's 2 M's. My name ends with an M and then microbiology begins with an M. And then go ahead and email those results. Now, I will be checking on a weekly basis this inbox to see the quiz reports. And salmmarianuniversity.edu. I'm also sending a copy to myself just in case there's any sort of a weird cyberspace glitch and I don't receive it. If I were a student, I would want a backup, obviously, so I don't have to go ahead and rework this assessment. So I'm going to email those results. Oops, so I need to also put my name. This is important because your name needs to be attached to the report. So don't put my name as the instructor, put your name. Section 1. Email the results, and this way when I get a hold of your report, I'm going to go back to this environment. When I get a hold of your report, you'll see here that your name is going to be coming up, and that's important that your name is attached to this report. Otherwise, for all I know, you're just sorry, I like to think the best of it, but if I were a student, you know, any number of weird things students will try to do to manipulate, homework assignments. So if I open up, this is the course Cell Microbiology inbox, I can see also the report is here. So this will be a graded metric that I'll actually enter into the gradebook. And so I'd like you to give that a try. Now, again, you can reattempt. Let's say you're not happy with this score. I would say go ahead and go somewhere else, and then come back in and see it's all blank again, so you can actually reattempt. So I want you to go ahead and complete the quizzes for chapters 1 through 4. You can see chapter 2 here has 30 questions as well. So these questions, again, are meant to sort of walk you through the reading material and make sure that you're not missing stuff that's important. And also the exams will be built off of questions that are modeled after these questions in your quizzes. So it gives you a chance to sort of, coach yourself to the exam, to what you can expect on the exam. All right. So we've got these links up at the top. Use the freeware and complete chapters 1 through 4. I'd like you to turn those in by Sunday. But if you can't, I'm sorry, not this Sunday because it's a holiday weekend. I'm actually extending that due date to the following Sunday, so we can talk about this more next week. Your research portfolio topics and refs also should be turned in, so let me go ahead and click on that. And here's what I've built into that Dropbox. So we have submitting to the Dropbox. I'm making this due actually by next Tuesday's class, or either electronically or in hard copy. I need you to take 8 ideologies, 1 from each column in that research portfolio that I gave you, that handout yesterday in class. I also need you to list 5 references in APA format. The references, no wiki, and then only 2 maximum can be a web page, and the web page must be a .org, .edu, .gov, or .net. Here is my email address and my cell number. If you have any questions or problems, you can go ahead and contact me. And yeah, this due date is going to have to be adjusted. How do you submit to the Dropbox, though? Let me go ahead and show you how you do that. So if I'm going to switch my role, let me go back here and show you a student view. Let me become a student here. I really should have been a student all along anyway. So when you go ahead and click on this, you have, I think, an opportunity to add a submission. When you click add a submission, you can go ahead and grab a file from anywhere. Let's just go ahead and grab a photo and drag it into the, ooh, that might have been too big though to submit. Oh, maybe not. Okay, cool. So then I went ahead and dropped my document into the Dropbox, and then I'm going to save the change, I think. Okay. Once you save the change, then you should be able to see that document in the Dropbox. So you don't have to worry about, is it there? I'm not sure. You should be able to actually see that file that's been submitted. Now, another option would be to go ahead and, oh no, I guess that really is your only option. So once it's submitted, you're good to go. And then you also have a practice quiz report Dropbox. So if you would prefer to not email it, as I have dictated here, you can go ahead and email it to cellmicrobiology@gmail.com. If you prefer actually to take that attachment and submit it in a document to this Dropbox, you can also do that as well. Not necessary, but there are a couple different ways that you can submit them. Either one is perfectly secure. So let me go ahead and get back to the course shell. You also have in Marian Online 2 the emailing option. And when you go into the emailing option, you have a list of all of your classmates. So if we do any kind of group work, or if you have a classmate that you want to communicate with, or with me, you can go ahead and build an email and then attach documents as well and submit. So that's good. And then you also have the option to view history. So if you want to see the history of emails, you can go ahead and do that. Now I think this is not sent yet. This is just a draft. that I'm going to be sending you shortly. All right. So you've got the course shell that talks about the objectives of the course and the assignments. This form is going to be changed to the chapter quizzes, the chapter quiz reports, I'm sorry, specifically. And then your research topics are going to need to be handed in. Next week, I'm going to actually build in those objectives. No, I'm sorry. I guess this is probably pretty complete. There's a couple of screencasts. So you see here, we have the research portfolio instructions that I handed out in hard copy yesterday. You have a screencast, a video that you can watch. When you open this up, it brings you to a YouTube vid that I've built. And again, I want to emphasize that you don't need to pay attention so much. This is the cover of the text that I used last time I offered this course. Now we've got a new text. So try when you're watching these screencasts to just filter out any references to chapters and anything that would be like text specific. Look for content. The content is the same. I'm trying to keep the content delivery relatively similar. We're just changing textbooks. I also want to draw your attention to this prokaryotic concept map. This concept map I've actually tooled to my courses. And if it's on this concept map, it's something you're going to need to know. So it may seem a little confusing now, depending on how much of the material that you've actually read through for the course. But you can see this Excel spreadsheet has 3 tabs. There's actually 3 different sheets in this document. The first sheet is abbreviated GPB and GNB. So this stands for gram-positive bacilli and gram-negative bacilli. This concept map is actually color-coded. So a gram-negative bacilli, for microbiology purposes, is going to stain pink. And I'm going to demonstrate a gram stain for you next week to give you a better idea of what these stains are really referring to. Gram-negative bacilli are going to be organisms that are going to be oblong, and they're going to stain pink. And we do some tests to sort of figure out what the genus and species is for that gram-negative bacilli. So this concept map says, okay, based on the way it's growing on the agar, we can tell whether it's utilizing lactose or not in a particular kind of medium called McConkey medium. If it's lactose fermenting, it's going to appear pink on this agar. And if it's pink and lactose fermenting, we go down this road to the yes. And then we do a test for indole. Indole production means that when we put the organism in touch with this reagent, there will be a blue color that develops. If it's indole positive, then we call it E. coli. So what we call here this concept map, it's something we're going to go over for the entire 8 weeks. This concept map is really a if this, then that, sort of key that we use to identify an organism that's infecting a patient. So once we grow the organism up on an agar medium, a solid substrate, we'll go ahead and put it through these series of tests. This yes-no sort of path will ensue until we get a genus and a species level identification. So for gram-negative bacilli, we have this dichotomous key. And these organisms on the bottom are going to be organisms that you're going to want to know about. So I would recommend making flashcards to study these. There's also a dichotomous key for gram-positive bacilli. So we're basically separating our organisms by color and shape at this point. And then all of these bottom squares here, Clostridium, Bacillus, we have, let's see, Listeria, Bacillus, again over here, Corynebacterium, Lactobacillus. If you click on this GNC tab, this will be gram-negative cocci. So we have down here Neisseриas and Moraxellas. And then for GPC, it has its own. So I would recommend that you make flashcards according to this prokaryotic concept map. And we'll see you next week. If you have any questions, don't hesitate to email, text, or call. Thank you very much, and peace out.